There are truths that science hides in plain sight, fragments of research published in sterile journals, wrapped in polite language and then quietly forgotten. But beneath the surface of modern medicine lies something deeply unsettling, something humanity has always whispered about in myths and legends, the merging of human and animal. Once thought impossible, now undeniable. Scientists have already created human-animal embryos. They admit it. They tell us they destroyed them before birth. But why create them at all? And what secrets are buried in the experiments they don't talk about? Tonight, we dive into the forbidden world of human-animal hybrids, a place where science fiction becomes science fact, and where humanity's future collides with its deepest fears. A chimera, in scientific terms, is an organism that contains cells from two genetically distinct organisms. Scientists have been creating animal-animal chimeras for decades, but human-animal chimeras represent an entirely new frontier, one that promises revolutionary medical breakthroughs and raises profound ethical questions. The process begins with precision that would have seemed like science fiction just decades ago. Human stem cells, master cells capable of becoming any type of tissue, are injected into animal embryos at their earliest stages of development. These aren't random experiments. They're carefully orchestrated attempts to solve one of medicine's greatest challenges, the critical shortage of organs for transplantation. The most publicized experiments have involved injecting human stem cells into pig and sheep embryos. The goal isn't to create some sort of mythological creature. The goal is to grow human organs inside these animals, hearts, kidneys, livers, that could save human lives. In 2017, Dr. Juan Carlos Izpisua Belmonte at the Salk Institute made headlines when his team successfully created the first human-pig chimeras. The embryos contained human cells integrated into pig tissue. It was a breakthrough that opened doors to possibilities that had existed only in imagination. The process relies on cutting-edge gene editing technology. Scientists use CRISPR to disable the genes responsible for organ development in the host embryo, creating a biological vacuum. Then, they introduce human stem cells, hoping they will fill that developmental niche and grow human organs within the animal's body. The results of these early experiments were both promising and humbling. After 4 weeks of development, the chimeric embryos contained approximately 1 human cell for every 100,000 pig cells, far from the millions needed for organ development, but proof that the concept could work. It was like planting a single seed in a vast field and watching it take root. But the challenges are immense. Human and animal cells develop at different rates, have different nutritional needs, and respond differently to growth signals. It's like trying to conduct an orchestra where every musician is playing a different piece of music, yet somehow expecting harmony. The research isn't limited to the United States. In Japan, Dr. Hiromitsu Nakauchi received approval to grow human-animal chimeras to full term, a controversial decision that pushed the boundaries further than any other nation. In China, researchers have created monkey-human chimeras. The United Kingdom allows chimera research under strict guidelines that require destruction of embryos after 14 days. Each country approaches the research differently, reflecting varying cultural, religious, and ethical perspectives on what constitutes acceptable science. What's forbidden in one nation is routine research in another. Early experiments revealed unexpected complications that scientists hadn't anticipated. Human cells often failed to integrate properly into animal tissues. They were outcompeted by faster-growing animal cells, or triggered immune responses that destroyed the embryo entirely. The efficiency remains extremely low, success rates measured in fractions of percentages. The vast majority of these experiments result in embryos that are not viable. Yet there have been breakthrough moments that have electrified the scientific community. In 2019, researchers successfully created monkey-human chimeras that survived for 19 days, longer than any previous attempt. The embryos showed remarkable integration of human cells into developing monkey tissue. Even more striking was a 2021 experiment that created human-monkey brain organoids. Human neural stem cells integrated into developing monkey brain tissue and contributed to cognitive development. The implications were both exciting and deeply troubling. For the first time, scientists had created organisms with hybrid brains containing both human and animal neural tissue. Today, dozens of research teams worldwide are pursuing chimera research. The focus has expanded beyond organ growth to include studies of human disease, drug testing, and basic developmental biology. These chimeras are helping scientists understand human genetic diseases, test new medications, and study how human cells behave in living systems. Some researchers envision disease models, where human diseases can be studied in animal bodies. Others see potential for testing drugs more effectively than current methods allow. The applications seem limited only by scientific imagination and ethical constraints. But with each breakthrough comes a new set of questions. As the science advances, the ethical complexities multiply exponentially. With great scientific potential comes profound ethical questions, questions that strike at the heart of what it means to be human. The moment scientists began mixing human and animal genetic material, they entered uncharted ethical territory, where traditional moral frameworks struggled to provide clear guidance. Human dignity is a cornerstone of medical ethics, embedded in every major declaration of medical principles since World War II. But what happens to that dignity when human cells exist within an animal body? Does the presence of human neurons in an animal brain grant that animal some form of human-like consciousness? These aren't abstract philosophical questions. They're practical concerns that researchers face every day. Religious leaders have voiced strong concerns across all major faiths. The Catholic Church has called chimera research a violation of human dignity, arguing that human life possesses inherent worth that cannot be instrumentalized for medical purposes. Islamic scholars debate whether such creations have souls, and if so, what obligations we have toward them. Buddhist philosophers question the karmic implications of mixing species, wondering whether such acts disrupt the natural order of existence. Perhaps the most troubling scenario is the possibility of creating animals with human-like cognitive abilities. Current research protocols require destroying embryos before they develop advanced neural structures. But as technology advances, this becomes increasingly complex. What if scientists accidentally create a pig with human-level intelligence? What are our obligations to such a creature? The question of consciousness haunts chimera research. We have ethical guidelines about experimenting on humans precisely because of our cognitive abilities, our capacity for suffering, for understanding our situation, for having hopes and fears about the future. If scientists create animals with similar capacities, don't those same ethical protections apply? This leads to what philosophers call the moral status problem. Traditional ethics assumes clear boundaries between humans and animals, with different moral obligations to each. Chimeras blur these boundaries in ways that our ethical systems weren't designed to handle. Governments worldwide are struggling to create appropriate regulations, and the rules vary dramatically from country to country. In the United States, federal funding for chimera research was banned from 2015 to 2016, following concerns about creating organisms with human consciousness. The National Institutes of Health now allows limited research with strict oversight, but private funding continues with fewer restrictions. This regulatory patchwork creates significant problems. Researchers can simply move their work to countries with more permissive laws. We need international coordination, but achieving that is politically complex when different cultures have fundamentally different views on the sanctity of life and the boundaries of acceptable research. Many ethicists worry about the slippery slope. If we accept human-animal chimeras for organ growth, what's to stop us from creating them for other purposes? Enhanced soldiers with animal strength, servants with reduced cognitive capacity? The possibilities are as limitless as they are disturbing. Science fiction has long explored these themes, from H.G. Wells' The Island of Dr. Moreau to modern films about genetic engineering. But these are no longer fiction. The tools exist today to fundamentally alter the nature of life itself, and the only barriers are legal and ethical constraints that vary widely across the globe. Yet, for patients dying on organ waiting lists, the ethical calculations look different. Every year, over 17,000 Americans are added to organ transplant waiting lists, while only about 39,000 transplants are performed. The gap between supply and demand grows wider every year, and thousands of people die waiting for organs that never come. These patients and their families often view chimera research not as an ethical problem, but as a potential solution to desperate circumstances. When your child is dying from organ failure, the abstract philosophical concerns about human dignity may seem less important than the concrete possibility of saving a life. The tension between individual medical need and broader ethical concerns creates one of the most challenging aspects of the chimera debate. How do we weigh the immediate suffering of patients against the long-term implications for society? How do we balance compassion for the sick with concern for the unknown consequences of crossing fundamental biological boundaries? Some bioethicists argue that the current focus on destroying embryos before they develop complex structures provides an ethical safeguard. By ensuring that chimeras never develop consciousness or the capacity for suffering, we avoid the most troubling moral dilemmas. But this approach may also limit the research's potential benefits. Others contend that any mixing of human and animal genetic material crosses a line that shouldn't be crossed, regardless of the potential benefits. They argue that human dignity requires treating human genetic material as sacred, not as a tool for medical research. The debate reflects deeper philosophical divisions about the nature of humanity itself. Are we simply biological organisms, different from animals only in degree? Or do we possess something qualitatively different, a soul, consciousness, moral agency, that sets us apart from the rest of the natural world? These questions become more pressing as the science advances and the possibilities expand. As we look toward the future, the questions surrounding chimera research become even more complex and the potential implications more far-reaching. What kind of world are we creating? And are we prepared for the consequences of our scientific ambitions? Technology is advancing at an exponential pace. CRISPR gene editing becomes more precise every year, allowing scientists to make increasingly sophisticated modifications to genetic material. Our ability to control cellular development improves constantly. What seems impossible today may be routine in a decade. Some researchers predict that within 20 years, we could have organ farms, facilities where animals are engineered specifically to grow human organs. Pigs could be modified to produce human hearts, sheep to grow human livers, primates to develop human kidneys. It's a vision that could eliminate organ shortages entirely, but it also raises profound questions about our relationship with the animal kingdom. Others envision more radical applications. Chimera research could lead to extending human lifespan by replacing failing organs throughout life. It might enable us to cure genetic diseases by growing healthy tissue to replace damaged cells. Some even speculate about enhancing human capabilities by incorporating beneficial traits from other species. But not everyone believes chimeras are the answer. Alternative approaches are being developed that might achieve the same goals without the ethical complications. Three-dimensional bioprinting uses living cells to print organs layer by layer. Tissue engineering grows organs from a patient's own cells on artificial scaffolds. Synthetic biology creates entirely artificial biological systems that could replace damaged organs. These technologies offer the promise of personalized medicine without the moral complexities of mixing human and animal cells. Organs grown from a patient's own cells would never be rejected by their immune system and wouldn't require a lifetime of immunosuppressive drugs. However, these alternatives are still decades away from clinical application, and their own challenges are substantial. Patients are dying now, while scientists debate which approach is most ethical and most practical. The question becomes whether we can afford to wait for perfect solutions when imperfect ones might save lives today. The societal implications extend far beyond medicine. How will chimera research affect our relationship with animals? If animals can carry human organs, does that change their moral status? Should we treat organ-producing pigs differently than regular pigs? Do they deserve special protection or consideration? Conversely, if humans can be enhanced with animal characteristics, does that change our moral status? If someone receives an organ grown in an animal, are they somehow less human? These questions may seem absurd now, but they could become pressing issues as the technology advances.
The research also raises questions about human identity and the nature of the self. If your heart was grown in a pig, is it truly your heart? If your brain contains cells that were once part of an animal, does that change who you are? Philosophy has grappled with questions of personal identity for millennia, but chimera research gives them new urgency and complexity. We need global governance structures to address these challenges. This research affects all of humanity, yet it's currently governed by a patchwork of national regulations that vary dramatically. The decisions being made in laboratories today will shape the future of our species, yet there's no international body with the authority to coordinate policy or ensure responsible development. Some experts call for a moratorium on advanced chimera research until international agreements can be established. They argue that we need time to develop ethical frameworks and governance structures before the technology advances too far. Others argue that delays could cost lives and that science doesn't wait for consensus. The next generation of scientists is being trained in an environment where these ethical questions are central to their education. Unlike previous generations, they're learning about the moral implications of their work from the beginning of their careers. They understand that every experiment has ethical dimensions and that scientific progress must be balanced with social responsibility. Yet they also see the tremendous potential to eliminate suffering and enhance human welfare. They're motivated by the possibility of curing diseases, extending life, and improving the human condition. The tension between scientific ambition and ethical constraint will define their careers and shape the future of biological research. International cooperation is essential but challenging to achieve. Different cultures have fundamentally different views on the sanctity of life, the relationship between humans and animals, and the appropriate boundaries of scientific research. What seems ethically acceptable in one society may be abhorrent in another. The pace of scientific advancement is outstripping our ability to develop appropriate governance structures. By the time international agreements are negotiated and implemented, the science may have advanced so far that the agreements are obsolete. We're trying to regulate a rapidly moving target with slowly moving institutions. Private funding adds another layer of complexity. While government-funded research can be controlled through funding restrictions, private companies and wealthy individuals can fund research in jurisdictions with permissive regulations. The democratization of biotechnology means that sophisticated research can be conducted with relatively modest resources. The future of chimera research may ultimately be determined not by ethical committees or government regulations, but by market forces and individual choices. If the technology proves safe and effective, demand from patients may override ethical objections. If it fails to deliver on its promises or produces unexpected consequences, it may be abandoned regardless of its theoretical potential. The creation of human-animal chimeras represents more than a scientific achievement. It represents a fundamental crossing of lines that have defined life on Earth for millions of years. We stand at a moment in history where the boundaries between species, between natural and artificial, between what is and what could be, are becoming increasingly blurred. Every transformative technology forces us to re-examine our values, our definitions, our understanding of what's possible and what's permissible. The printing press challenged religious authority. The Industrial Revolution transformed our relationship with labor and nature. Nuclear weapons forced us to confront the possibility of human extinction. Chimera research is no different. It challenges fundamental assumptions about life, identity, and human nature. The embryos created in these experiments exist for only days or weeks before being destroyed. They never develop into complex organisms, never breathe, never experience consciousness. Yet the questions they raise will persist long after they're gone. Each experiment pushes us further into uncharted territory, where our moral compasses struggle to find direction.
We stand at a crossroads where multiple paths diverge into uncertain futures. Down one path lies the potential to eliminate organ shortages, cure genetic diseases, and extend human life. It's a future where death from organ failure becomes as rare as death from infection is today. Where genetic diseases that have plagued humanity for millennia are eradicated. Where the boundaries of human capability are expanded beyond current imagination. Down another path lies the risk of unintended consequences we can't yet imagine. The possibility of creating new forms of life that don't fit into our existing moral categories. The danger of reducing human life to its component parts, to be manipulated and instrumentalized for utilitarian purposes. The potential for exacerbating existing inequalities by creating biological advantages available only to the wealthy. The choice isn't whether to pursue this research. It's already happening around the world in dozens of laboratories. The choice is how to pursue it responsibly, how to balance the promise of medical breakthroughs with respect for human dignity, how to address the urgent needs of patients while considering the long-term implications for society, how to proceed with hope while acknowledging uncertainty. We need robust public dialogue that includes not just scientists and ethicists, but patients, religious leaders, philosophers, and ordinary citizens who will be affected by these decisions. We need international cooperation that respects cultural differences while establishing common standards for responsible research. We need ethical frameworks that can evolve with the technology while maintaining core principles about human dignity and the value of life. The decisions we make today will determine whether chimera research becomes humanity's greatest achievement or its greatest mistake. Whether it represents the triumph of human ingenuity over biological limitations or the dangerous hubris of a species that has forgotten its place in the natural order. In laboratories around the world, scientists continue their work, creating embryos that blur the line between human and animal, then destroying them before they can develop into complex organisms. Each experiment brings us closer to medical breakthroughs that could save millions of lives. Each also brings us closer to questions that may redefine what it means to be human. The lines have been crossed. The boundaries that once seemed fixed and permanent are proving to be malleable and temporary. There's no going back to a world where human and animal remained strictly separate categories. The question now is not whether we should have crossed these lines, but where we go from here. We are writing the next chapter of human evolution, not through natural selection, but through conscious choice. The responsibility is enormous. The stakes could not be higher, and the outcome remains uncertain. But one thing is clear. We can no longer pretend that the natural order is immutable or that the boundaries of life are fixed. The future of chimera research will be determined by how well we balance scientific ambition with ethical wisdom, medical need with moral principle, hope with humility. It will test our capacity for collective decision-making on issues that affect the entire human species. The chimeras themselves may exist for only days or weeks, but their legacy will endure for generations. They represent both our greatest hopes and our deepest fears about the power of science to transform life itself. The question is not whether we have the power to cross these fundamental boundaries. We do. The question is whether we have the wisdom to wield that power responsibly. Time will tell whether we are witnessing the dawn of a new medical age or the twilight of human dignity. Whether we are solving the greatest challenges facing humanity or creating new ones we can't yet comprehend. The only certainty is that there is no turning back. The lines have been crossed, and we must live with the consequences of our choices. The story of human-animal chimeras is still being written. Each new experiment adds another page, each ethical debate another chapter. We are all characters in this story, whether we realize it or not, and its ending remains unwritten.